You know, every single second, your body is performing an absolute miracle. Trillions of times over. It's building the very code of life, basically from scratch. Today, we're going to pull back the curtain on how this incredible process works, and, even cooler, how hijacking it is helping us fight some of our most challenging diseases. Let's dive right in.
So that's the big question, right? How in the world do our cells take these simple, common molecules floating around and assemble them into the A's, T's, C's, and G's that write the instruction manual for everything that you are? It is a biological construction project of unbelievable precision, and it's happening inside you right now.
And believe me, understanding this isn't just for biology nerds. This exact process is a major battleground in the war on cancer. It's also absolutely critical for keeping our own immune system in check, helping to prevent the rejection of life-saving organ transplants. And when the super intricate assembly line gets gummed up, it can lead to some seriously painful conditions like gout. So yeah, the connection to our health, it's about as straight as it gets.
Okay, so before we can really get into how medicine throws a wrench in this system, we first have to meet the key players, the actual ingredients our cells are working with.
Everything, and I mean everything, starts with this little guy, the nucleotide. The easiest way to think about it is like a three-part Lego brick. You've got the nitrogenous base, that's the letter like A, T, C, or G. Then you have a pentose sugar, which forms the backbone. And finally, a phosphate group that kind of snaps it all together. Our cells have to build all three of these parts. Now, these bases aren't all created equal. They actually fall into two different families. On one side, you've got the pyrimidines, which have a simpler single ring structure. And on the other, you have the purines, which are bigger with a double ring. And this little difference in their shape, it's a huge deal because it means the cell needs two totally different assembly lines to build them.
So let's start with the simpler crew, the single ring bases. We're talking about the pyrimidines, cytosine, uracil, and thymine.
The strategy for building pyrimidines is pretty straightforward. The cell builds the nitrogenous ring first. It just takes some basic ingredients and snaps together this core ring structure. It's only after that ring is totally finished that it attaches a pre-made sugar phosphate backbone, something called PRPP, to complete the nucleotide. It's kind of like building a car's chassis first and then just dropping the engine in at the end. All right, now for the other assembly line. This one handles the bigger, more complex, double-ringed purines, adenine and guanine. And this is where things get wild, because the pyrimidine strategy is the complete opposite. Here, the cell starts with the sugar phosphate foundation and then builds that complex double-ring structure piece by painful piece directly onto that foundation. So it's less like dropping an engine in and a whole lot more like building the engine from scratch, bolt by bolt, right on the chassis. And because this ring is built piece by piece, you can imagine it requires a much more diverse shopping list of ingredients. We're talking glycine, formyl THF, glutamine, CO2, aspartate, the whole shebang. It's a complex recipe, but it all leads to one really important molecule, which we're about to see. So the whole flow of the process is just different. It kicks off with that sugar foundation. Then, in a pretty complicated 10-step process, it adds all those ingredients we just saw. And the entire goal of all 10 of those steps is to create one single crucial molecule, inosine monophosphate, or just IMP. You should think of IMP as a central hub or a fork in the road. From that one molecule, the pathway can branch off, allowing the cell to turn IMP into either adenine or guanine, whichever one it needs at the moment. So what we have here are these two incredibly intricate, high-demand production lines running non-stop in our bodies. And this brings us right back to where we started, modern medicine. Because when a process is this essential to life, well, it also makes it a perfect target.
And it's no joke. The source material spells it out pretty clearly. When the regulation of these pathways goes haywire, the consequences are severe. And this vulnerability, this dependence on the assembly line, is exactly what scientists have figured out how to exploit with drugs.
And just look at how incredibly specific these drugs can be. Take mycophenolate. It targets the purine pathway by blocking the exact enzyme that turns that IMP hub into guanine. No guanine, no new DNA for those out-of-control immune cells. And then you've got drugs like 6-mercaptopurine, which is basically like throwing a whole bunch of wrenches into multiple gears of the purine assembly line at once, grinding the whole thing to a halt.
The strategy behind all of these drugs is actually pretty simple and pretty brutal. It's starvation. See, cells that are dividing like crazy, we're talking cancer cells or the immune cells that cause organ rejection, they have a massive, insatiable demand for new nucleotides. So by cutting off their supply of these essential building blocks, these drugs effectively starve them, slowing them down or stopping their growth altogether.
And when I say the demand is immense, it is truly mind-boggling. Your body has to create somewhere in the neighborhood of 100 billion new cells every single day. And every one of those cells needs a complete, perfect copy of your DNA. That is the staggering scale of the manufacturing challenge our cells solve second by second. And that kind of leaves us with one final, big thought to chew on. We've become incredibly good at turning these pathways off to fight disease. But what's next? What could we possibly achieve in fields like regenerative medicine if we learned not just how to sabotage the assembly line, but how to safely and precisely turn it back on? Now that is the next frontier. Thanks for diving in with me on this one.